Welcome to the Exam Technique series on the Science Revision Coach channel. This series is designed to support you with your exam technique so that you can show off the science knowledge and skills you have been able to recall from the Revise Science with Me series. This video is about how to answer questions on photosynthesis. Before we can attempt past paper exam questions on this topic, you first need to be aware of how to approach exam questions to ensure that you can use the time allocated efficiently. So for those of you who've already heard it, it's just a good reminder. For those of you new to this series, please listen carefully. Step 1, it sounds obvious, but read the blurb of the exam question. This sets the scene of the question and gives you an opportunity to think about whether you are confident to answer the question straight away or whether you will need to come back to it. If you are confident, move on to step 2. If you are not, this is the time that you need to move on to the next question and follow the steps above. Step 2, deep read the blurb, data, and any diagrams. Do you understand what they are telling you? Have you noticed any patterns or anomalies? Highlight or underline any information of note and annotate as appropriate any of the key information that you have noticed. Step 3, read the actual question. The question would have a command word in it, and if you're not familiar with the types of command words used in GCSE science exams, you can see the link on the sciencerevisioncoach.org site on the students page for a comprehensive list of AQA command words. Step 4, using your annotations and your knowledge and understanding, begin to answer the question. Remember, you do not have to write in detailed essays for extended response type questions. You can write using clear bullet points, labeled diagrams, tables, and flowcharts. It is advisable to use a sharp pencil and a ruler to plot and draw graphs. Using a ruler for accurate measuring of data on graphs is also advisable. Use black pen to write your responses using clear scientific language and legible handwriting. Lastly, don't forget that answers from previous parts of the question may be needed in later parts of the question. So let us have a look at this question that I have on the screen. Remember, 1 mark is roughly 64 seconds because in the science exams, you have 70 marks in 75 minutes. This is an 8-mark question, so it took just over 8 minutes to complete. You can put a stop clock on to time how long it takes for us to get through this question using all the exam approach steps that I've just advised, just so that you can see that what I'm saying is true. You do need to spend the time highlighting, annotating the information provided to you before you can answer the question to ensure that you use your time effectively and you don't make silly mistakes. So you can start it now. So this question shows me that it is about the photosynthesis required practical. I have annotated the blurb and the diagram with what I remember about the practical, and I focus on the independent variable, the dependent variable, and the control variables. I always remember more than 2 or at least 2 control variables for every required practical. I also have listed any equipment or materials that are not included in the diagram. I have also included the balance symbol equation, and this will help me to remember the products produced, and that is actually the first question. The next question, part B, is asking me to describe one way the student could change the intensity of the light reaching the pondweed. And I've already annotated my blurb of the question saying that distance from the lamp source is the independent variable. So my answer is going to be basically moving that light source to different distances away from the pondweed. The next part of the question, part C, is asking me to describe how the student could use the apparatus in figure 1 to measure the rate of photosynthesis. So rate is basically how quickly something is done or happens over a period of time. So I have used the information that I have written from my annotations, basically the dependent variable, which is to count the number of bubbles released every minute at each distance the lamp is moved from the pondweed. The next part is a graph showing the students' results. And I've annotated the graph looking at how much each of the tiny squares is worth. Um, I've looked at where light intensity is a limiting factor and where carbon dioxide could be a limiting factor, for example. And the question for part D says, what was the maximum rate of photosynthesis and what was the highest point of the graph? And that was 34. And I used the ruler to accurately measure this and make sure that I make a note of how many tiny squares is equal to one arbitrary unit in terms of the rate of photosynthesis. Part E is a multiple choice question, and I've got a choice between 200 lumens, 600 lumen, and 1200 lumens, for which part of the graph shows that light intensity was a limiting factor. Now, because I had already annotated my graph, it could only be one value, which is 200 lumens. For part F, light intensity affects the rate of photosynthesis. It says give one other factor that can affect the rate of photosynthesis. And the first thing that came to my mind was temperature. It could have also been carbon dioxide concentration. But, I write down the first thing that comes to mind. And as you can see for these questions, I'm getting them all right. So now let's look at the examiner's report. Obviously, check the timer, and hopefully you'll see that we're well within the time to answer the questions that we've had. For question 1A, over half of the students correctly named the gas that is produced when the pondweed is in the light as oxygen. When students are asked for the name of a chemical, they should write the name, not give the formula. If an incorrect formula is given, for example, O with a big 2, the letter O on its own, or O with a superscript 2, the mark cannot be awarded. So you're giving yourself more chance to make an error. If they say name, write the name in English, not a chemical symbol. If they ask for the formula or the symbol, then you do what they've asked. For part B, about 58% of students gained 2 marks for saying the light intensity could be changed by moving a lamp nearer to or further from the pondweed. Some suggested altering the temperature, using more pondweed, or leaving it for a longer period of time, and none of these gained credit. For part C, half the students gained 4 marks for this question, which differentiated between students very well. So this could be your grade 9-8 question, if you like. It was very clear when students had a real practical grasp of the investigation. They gave a concise response describing counting the number of bubbles of gas produced in a given time or in 1 minute. When only 1 mark was awarded, this was usually for the first marking point, to count the bubbles. Other students described how to vary the light intensity rather than describing what should be measured in order to calculate a rate. So they were getting confused between the independent variable and the dependent variable. For part D, around 83% of students correctly gave the maximum rate of photosynthesis as 34 arbitrary units. As usual, for graph readings, a tolerance of plus or minus half a small square was allowed. So it's a very, very small square. So make sure you use a ruler to make sure that this is accurate. Therefore, a value between 33.5 and 34.5 was allowed. Incorrect readings of 35 and 40 were commonly seen. So this is where students have been typically lazy. They just have kind of gone, oh yeah, it's about 34. Oh, it's about 35. Or students who are not wearing their glasses if they need to have glasses. So please make sure you're really checking. Some students read the wrong axis and gave an answer of either 600 or 1200. So check that carefully. And this is why it's so important to annotate graphs so you understand what they're telling you. For part E, about 12% of students identified the correct part of the graph when light intensity was limiting the rate of photosynthesis. The vast majority selected 600 lumens as the answer. For part F, the question asked for a factor that affects the rate of photosynthesis. Many students gave light intensity, which had been given in the question and was therefore ignored. Around 64% of students answered correctly, with the most common correct response being temperature. The amount of carbon dioxide and water availability were other correct responses that were seen. So, as you can see, you might want to pause the video just to reflect on what you've seen and heard, because there was a lot of information about what students did well and what students didn't do well. So for the next question, which is worth in total 16 marks, you can start your timer now. The first part of the question uses the word photosynthesis. I need to complete a word equation, so I've underlined the fact that it's a word and not a symbol equation. So I write down what I remember from the word equation. And then it says light intensity affects the rate of photosynthesis. The diagram below shows some of the equipment used to measure the rate of photosynthesis. So again, I'm going to recall the practical, and I've written down some of the equipment that I can see is missing from the diagram. There's no stop clock, there's no ruler, for example. And it says describe a method to investigate the effect of light intensity on the rate of photosynthesis. Now, ordinarily, I would write down the independent variable, dependent variable, and control variables very quickly in my annotations. But because I've just done that literally in the last question, I haven't done it here, but that's what I would do if I was doing practice questions. So please don't follow my example in that respect. But you can see that it's a similar type of question. And this is why when you do repeated past questions, you get familiar, you're doing retrieval practice, it becomes embedded in your long-term memory. So I mentioned the independent variable and that I need to move the lamp to different distances from the pondweed, 5 different distances. I could improve that bit by actually mentioning the ruler. I mentioned the dependent variable and measuring the number of bubbles released every minute at each of the distances. I mentioned control variables such as keeping the pondweed the same, using a water bath to keep the temperature the same, and to control the carbon dioxide content in the water. I then talked about repeating the test at each distance. I've talked about calculating a mean number of bubbles and using the data to calculate the mean rate of photosynthesis at the different light intensities. And then when I look at the mark scheme, I can see that I would have got level 3, which is 6 marks. I need to mention what I'm changing to what I am measuring and what I'm controlling. That's what I need to do to get full marks in this type of question. For the next part of the question, it says algal cells photosynthesize. So I'm talking about another type of producer. It says scientists investigated the effect of light intensity on algal cells. The algal cells were placed at different light intensities, and the table below shows the number of extra algal cells after 2 days. So I look at the table and I can see that there is an increase as the light intensity in lux increases. The number of extra algal cells after 2 days also increases, but it does slow down towards the end. And then it says the initial number of algal cells was 200,000, and that I have to calculate the total number of algal cells after 2 days when the light intensity was 500 lux. So straight away, I'll go to the table to the 500 lux and I highlight the corresponding extra algal cells after those 2 days, and I write that down. And that was 1.65 times 10 to the power of 6. And I add that to my 200,000, which I have also converted into standard form just to make it easier. Less chance of me leaving off a 0. And I calculate the sum of that and I get my answer, which is 1.85 times 10 to the power of 6 cells. And obviously checking the mark scheme, I can see that I've got that right. For the next part of the question, part D, I have to plot data from the table onto the graph. Now, because I've been able to manipulate this, I've been able to put the table onto the same screen as the graph. And obviously in the exam, you would have the table and graph on the same double page spread, so don't need to worry about flicking back and forth between pages. You can see that I've ticked off what plots I have done to make sure that I haven't missed anything out. That's just good practice. And you can see that I have used crosses, I call them St. Andrew's crosses, to make it clear to the examiner where my plots are, because the examiner will check every single plot. I know this because I'm an examiner myself. And as you can see, when I draw my line of best fit, if I do dots or tiny circles, you might not be able to see the plots at all once you've done your line of best fit. Now, again, just like with the previous graph question on another video, I have drawn this on a graphics tablet, so it's really hard for me to get a smooth, continuous line, but it's the best I could do after several attempts on the graphics tablet. And on paper, I would be able to draw a lot smoother of a curve. But the point that I wanted you to see is that I haven't got artistic shading. I have got one smooth, continuous line, or relatively smooth, continuous line through most of my plots. And therefore, I've got the 2 marks for the plots, and I'll get the mark for the line of best fit. For question part E, it says, give 2 conclusions from the results. Use information from the table above. And again, in an actual exam paper, this will all be on one double page spread. So you could keep an eye on the graph whilst you're answering the question. And I could see that as light intensity increases, the number of algal cells increases. That's first conclusion. And then the other thing I could see is that this did not go on forever. At 1250 lux, the number of algal cells plateaus. It does not get any higher. And then for part F, it says explain. So I've got to use the word because in my answer. Explain how an increase in temperature from 20 degrees Celsius to 25 degrees Celsius would affect the number of algal cells. Now, 20 to 25 is like a lovely warm spring day. Even you could almost argue that it's summer. So that's a lovely day. It's not beyond the optimum for most of the enzymes in photosynthesis to be denatured, so it's ideal. So I've put increase the number of cells because photosynthesis would happen at a faster rate because the enzymes involved would catalyze the reactions faster. Notice I've used the word because. Now, unfortunately, there was no examiner's report for that last question, but I was able to give you my own examiner's advice when answering a question when you have to plot a graph. And I think that you should consider that invaluable advice because many students throw away marks that are quite easy to get in terms of plotting data. The line of best fit takes practice. You don't have to be a fantastic artist to draw a line of best fit in science, but you do need to know that it needs to be a smooth, continuous line. And if the plots are plotted correctly, there may be one anomaly, but you can see if it's supposed to be a straight line. You can see if it's supposed to be a curve. And therefore, you use a ruler if it's a straight line. And if it's a curve, you do as best freehand, smooth, continuous line of best fit as possible. And that's what I wanted you to see from that particular question. Thank you for sticking around for this time. It's been less than 20 minutes, so perfect time for you to do some revision. You can access past papers from the website of your exam board or have a look at the description box below for more details of how you can access more exam papers. If you haven't already subscribed or pressed the like and notification buttons, please do so now for this channel. And if you need any further resources, check out the sciencerevisioncoach.org website for a whole host of resources. Good luck.